suspicion that she'd been exposed, but she luckily didn't have it. She turned out to be negative.
Okay, but she was vaccinated anyway, so most likely the
yeah, she was vaccinated.
Been so severe.
Yeah. That she'd been exposed.
My job is to make college easier because students have a lot of fun.
So we are live on YouTube now. So everything we say is gonna be public.
Okay.
Vote against us.
Exactly.
Somewhere I can also.
Shall we already let people in, Gilad?
I'm going to let them in now.
Mm.
I should be able to admit all of them together.
I think I just did.
Okay, we still have some time before we start.
Right. So Ben, where are you now, apart from being in a cafe? Which city are you?
I'm back to Switzerland. Returned from Denmark on Friday, and yeah, now I'm back here, and I have a meeting with the foundation on whose board I sit, and this is nearby, and so on the way I'm now here on the coast of Lake Zurich, and great to hear some
nice. But it's also stressful
from Peter. Pardon me?
So you're traveling back and forth now between Copenhagen and Zurich?
No, I'm my Copenhagen trip has ended.
Ah, right.
I'll be here for the time being.
That was a very short sabbatical.
It wasn't really a sabbatical. It was really just what was planned to be a 2-month stay, but then for reasons that you know, it got cut short.
Yeah. So Peter, are you planning to have any sabbatical, or it's not needed because you're anyway in California? What else could you wish?
I have never taken a sabbatical. There's so much going on here, and it's maybe one of these days I should.
Totally understandable. Yeah. Yeah, but travel restrictions sort of also step by step go down.
Yeah.
I'm traveling continuously between Europe and Israel, and that gets smoother and smoother.
Okay, yeah. I must confess I've not been on an airplane for 18 months. The last time was a meeting in Switzerland in January of last year, and there's just no meetings in the US.
Yeah.
So,
Oh yeah, that was Fred Holland's meeting, right?
that's right, yes.
Yeah.
So how does it feel to be not to be in a plane for 18 months?
It feels very strange.
What does he expect? It feels good because sitting on a plane is typically not the most likable thing.
I think it's just, you get more done actually staying at home and working all the time instead of traveling around.
That's true.
Yeah, that's probably true. Productivity can go up like that.
Unless
Yes, but
small kids running around.
yeah, but from November, apparently, we Europeans are again allowed to enter the US. So I'm sure many of us will start to come over again.
Yeah, things will open up again for sure.
Okay. We can wait a couple of more minutes.
Yeah, the numbers are
Still going up.
I see a number of good friends. Hello.
Hello, Helmut.
Helmut here. Hi.
Hi,
Hello,
how
hello. Hi, Helmut.
are you?
Great. Thanks. More and more traveling again.
Uh,
And I also see Dima. Hi, Dima.
Hi guys.
Hi, Dima.
Hagen, where are you? I don't recognize.
I'm back in my office.
Oh, that's your office. That's why. There's an office?
Looks like my living room, right?
That he went back to Israel.
Yeah, I'm back in Israel. Without quarantine this time.
Oh, nice.
Yeah. Yeah, I think if I accumulate all my quarantines that I had over the past year, I easily reach 2 to 2.5 months.
That's crazy.
Yeah, you really,
Means time you can work without interruption.
yeah, that's true. But you're also gaining a few kgs, so you get a big fan of dance aerobic. Numbers are still increasing, Kalas.
Yes. I see also Peter and Jose and Laila and other friends. Bill was somewhere there.
Looks like we're saturating now in terms of numbers.
Okay, so let's start. Hello, everybody. So we are back in the protein folding and dynamics webinar, and we hope to keep it at least for a while in the same format every 2 weeks. We're thinking whether to make it a little bit more sparse at some point or not, but we haven't decided yet. If you have any input, you can write to us about that. So we are very happy to have with us today Peter Wright from the Scripps Research Institute. I think I almost don't need to introduce Peter. Everybody surely knows him very well and his great work, but nevertheless, let me say a few words about Peter. Peter did his PhD in the University of Auckland in New Zealand. He postdoc in Oxford, and then in 1976, he joined the University of Sydney as a scientist. And in 1984, he moved to San Diego, I guess, or to La Jolla, I should say, to join the Scripps Research Institute. Peter has been doing various positions over the years, and people like him so much that they keep him in those positions forever. He was the chair of the molecular biology department at Scripps from 1987 to 2012. And he's been the editor-in-chief of the Journal of Molecular Biology since 1990 and still going very strong. Peter's research has focused on applications of nuclear magnetic resonance to study protein folding and dynamics, very much on the topic of this webinar, of course. Peter worked on the folding of several proteins and really solved the mechanisms of their folding. For example, apomyoglobin, which we heard in this series from Jane Dyson, and also transerythrin, which we've been studying in the last few years. And Peter has been also a pioneer in the IDP field. He was one of the first to realize that proteins can also exist without a structure, and he used sophisticated NMR methods in order to actually decipher the structure that they do have when they're unfolded, and then went on to understand how these unstructured proteins actually function in fairly sophisticated ways as part of the biology of cells. He looked at folding upon binding, and then he looked at networks of IDPs as they interact with each other, where one can influence the folding of the other, and then in that way behave in fairly sophisticated manner, like suppressing the activity of one protein binding by binding to it, and so on and so forth. And I'm sure that we are going to hear about some of this work today in the lecture. Peter is a member of the National Academy of Sciences for many years. He is also a fellow of the American Academy of Arts and Sciences. He received many awards, and I will just mention the Stein and Moore Award of the Protein Society in 2010, and the International Society of Magnetic Resonance Award in 2019. Just before giving the microphone to Peter, I would like to mention the way we run this talk, to remind you that I ask all of you to mute your microphone during the talk, and in the end of the talk, if you have a question, please write it in the chat, and I will ask you to unmute yourself one by one and ask the question in person. And we can stay as long as there are questions, and usually we have very nice discussion after the talk, so please stay with us for the discussion. So Peter, with that, I will stop and I will let you start the lecture, please.
Thanks, Gilad. So I'm going to talk to you today about protein disorder. This is, as Gilad said, this is something that we've been interested in for a very long time. We've spent a lot of effort in trying to understand the role of disordered proteins, how their dynamic properties, their unique properties, make them particularly well suited for certain biological functions. And today I'm going to talk about the role of disorder in regulation of cellular signaling, and particularly by the protein p53, the tumor suppressor p53. Now I seem to be having trouble advancing my slides. There we go. Okay. Yeah, so I'm sure I don't have to explain this to anybody in this audience, but intrinsically disordered proteins, IDPs, or intrinsically disordered regions, IDRs, are found in about 40% of proteins in the human proteome. These proteins are either entirely disordered or partially disordered, with regions of more than about 30 amino acids showing disorder. And again, the characteristic of IDPs is that in isolation, they're unable to fold spontaneously into well-defined, stable globular structures. And beyond the sort of 40% of proteins in the proteome being heavily disordered, it turns out that we now believe that most eukaryotic proteins contain both folded domains and disordered regions. And understanding these is going to be extremely important in understanding how these proteins function. Why is that not advancing? There we go. Okay, so to characterize IDPs, we want to understand the structural ensembles and the interactions that they make. And in the case of the full-length proteins that contain both ordered and disordered regions, this is going to be a major, major challenge. So most of the existing biophysical analysis of IDPs and their interactions has used a reductionist approach, where one dissects the protein, separates out the ordered and disordered regions, and then studies these in isolation. Now, this doesn't give us a complete picture of how these proteins function, because the domains don't function in isolation. They may have some of the properties of the particular protein, but they don't show how the domains synergize to function together. And so a major challenge now is to understand from a more holistic approach how the globular domains and the disordered regions function together. And there's no one technique that can give you all the information you want. I think this is going to require hybrid approaches. NMR is certainly playing a major role. And the approach that I'll talk about today is to use segmental labeling with NMR. One can use X-ray crystallography to get structures of the individual folded domains. Single molecule fluorescence plays a major role, SACs, et cetera, probably cryo-EM in some of the biggest systems in the future. So I want to emphasize that the disordered regions of protein are an integral part of it. They're not just evolutionary junk. They function alongside the globular regions to synergistically modulate the biological function. And I'll give you some examples of that today by talking about the role of the intrinsically disordered regions of p53, the tumor suppressor, in regulating its biological activity, particularly its DNA binding. So this slide shows you the domain organization of p53. And about half of the protein is unstructured, it's disordered. So the human p53 is 393 amino acids in length, and only about 200 of those amino acids are in structured regions in the DBD, and then a smaller number in the tetramerization domain. The N-terminal region, N-terminal 91 amino acids, is entirely disordered. It contains the N-terminal transcriptional activation domain, or NTAD, I'll call it for short, and a proline-rich domain. Within the N-terminal activation domain, there are two amphipathic motifs that mediate interactions with other proteins. In the disordered C-terminal region, there's a long disordered linker between the structured DBD, DNA binding domain, and the tetramerization domain. And then the last roughly 40 amino acids are entirely disordered, but they contain a C-terminal regulatory domain. I'll call that the CTD for short. In terms of structure, there are many, many structures, X-ray structures of the DNA binding domain, both in isolation and bound to DNA. There are structures of the tetramerization domain. So as a monomer is disordered, it folds into a tetramer and holds the p53 into its tetrameric state. The structures of the activation domain, it's entirely disordered with very little propensity, except in the AD1 region, for a small amount of helical structure. But when it binds to targets, it folds. And so one of the key properties of the activation domain is that through AD1, it interacts with the ubiquitin ligase MDM2, which leads to degradation of p53, or after phosphorylation and post-translational modification, MDM2 is displaced, and that N-terminal activation domain is then available to bind to transcriptional coactivators such as CBP and P300. And this shows a structure determined by one of my grad students a few years ago with the entire 61 residue activation domain bound to the TAS2 domain of CBP, the coactivator CBP. You can see that AD1 folds up into a helix, AD2 folds up into a helix, and there's some disorder and some structure in the region between AD1 and AD2. So p53 is activated and regulated by post-translational modifications, a huge number of post-translational modifications. And these are indicated on the slide. So you can see that in the NTAD, the internal activation domain, there are numerous phosphorylation sites. And these are involved in stabilization of p53, but if by phosphorylation of S15 and T18, MDM2 is released. And the phosphorylation in this region also enhances the binding affinity for the transcriptional coactivators, CBP and P300, that are required to activate transcription of p53-mediated genes. At the C-terminal, there are some phosphorylation sites, and there are numerous acetylation sites. And some of these acetylation sites, 372, 373, and 382, for example, are acetylated by the coactivators CBP and P300. So when CBP and P300 are recruited by interactions with the N-terminal activation domain, the histone acetyl transferase domain of CBP and P300 can acetylate the C-terminal regulatory region, the CTD. So these post-translational modifications play a critical role in the activity and regulation of p53. Now, one site of modification that is known to be biologically very important, T55, does not in any way affect binding to coactivators or to MDM2. So we were intrigued to know what the role of T55 phosphorylation is in the regulation of P53. Firstly, before I go on to talk about that, let me remind you that P53 DNA binding domain binds cooperatively to DNA as a tetramer. It's actually a dimer of dimers. So we have the green dimer here, the yellow dimer there, and there's cooperative interactions between the DVDs involved in the binding to DNA. P53 recognizes typically a 20 base pair consensus sequence, specific sequence, and that consists of two 10 base pair half sites. There's also interactions with DNA through the highly basic CTD region, the C-terminal domain. And the interactions of that are non-specific, whereas the DBD themselves make specific interactions, recognize specific DNA sequences. So we were interested in moving beyond a reductionist approach. We'd used a reductionist approach to look at the interactions of the N-terminal activation domain, for example. We wanted to look at the full-length protein to understand synergies between the disordered regions and the structured DNA binding domain. And so we undertook high-resolution NMR studies of the full-length P53 tetramer, which is about 180 kilodaltons in molecular weight. Now, we used a superstabilized P53 tetramer, introducing mutations that were identified by Alan Thirsch many years ago, which is 4 mutations, N133L, V203A, N239Y, and N268D. And these mutations have been introduced to stabilize the DBD. The DBD is not very stable. And so to stabilize it for NMR studies, we use these mutations. They have absolutely no effect on the properties of P53, and just make it easier to study by biophysical methods. So as I mentioned, the proteins that contain both globular domains and long disordered regions are a major challenge. And what I show you here is a TROSY spectrum of full-length P53 tetramer labeled with N15 and deuterium. And you can see that the central part of the spectrum is dominated by very strong resonances that come from the disordered regions. The resonances from the DBD are very weak, and a lot of them are missing. And we see no resonances at all from the tetramerization domain in this tetramer. And so the NMR is complicated by having this horribly overlapped spectrum with enormous difference in intensity between the signals of the disordered region, which are heavily overlapped in the middle, and the resonances from the DBD, which are very weak. This is a spectrum obtained by Josephine Ferrion in my lab many years ago. So to get around this problem, Alex Croyce, a grad student in the lab, undertook intine-based segmental labeling of the P53 tetramer. And with help from Maria Yamut, he was able to generate segmentally labeled P53, where either only the N-terminal activation domain was labeled, or the nuclear localization signal between the DBD and the tetramerization domain, the TET domain itself, and the CTD, the regulatory region, were labeled. So we could generate these two different constructs. So here's a uniformly labeled, 15N labeled P53, just focusing on that central region where the signals from all of the disordered regions appear. Here's the construct that's labeled only in the N-terminal activation domain. You can see it's a much simpler, beautiful spectrum, nicely resolved. And here's the spectrum of the NLS TET CTD region, again within the full-length P53 tetramer. There's some broadening there, and as I said before, we see no signals from the tetramerization domain, even though it is a folded entity. So using these approaches, Alex could obtain highly simplified spectra of full-length p53. So let me first talk about interactions with the C-terminal activation domain interacting with DNA. And so again, we're using the NTAD labeled p53, where just the C-terminal region is labeled, and we're seeing signals primarily from the NALS, in fact, entirely from the NALS and the CTD regulatory region. This is work done by Alex Croyce and Sang-Ho Park. And I emphasize again, it's full-length p53 tetramer. So the CTD makes transient and non-specific interactions with DNA. So we're interested to know, where are these interactions? Does it interact with DNA in the 20 base pair recognition element, or is it interacting with DNA outside that region? And so Alex collected data using DNAs of increasing lengths, just using a p21 consensus site, 20 base pair p21 consensus site. And if you look at the red and the black spectra here, you see that there's very little perturbation of chemical shift on binding of the full-length p53 TBD to a 20 base pair DNA. As he increased the length of DNA, adding base pairs outside the regulatory element, recognition element, then you can start to see chemical shift changes. So with a 60 base pair DNA, with 30 base pairs either side of the, in fact, 35 base pairs either side of the, 30 base pairs either side of the regulatory element, we start to see shifts of resonances. And he determined the average shift of all the resonances that are affected and plotted those versus the number of base pairs in the regions outside the recognition element. And you can see that as the number of base pairs outside the recognition element increases up to 30, we get a steady increase in the chemical shift perturbation. So this tells us that the CTD is not interacting in the recognition element itself, but it's anchoring the p53 to the DNA through interactions in flanking regions. Alex and Sang-Ho Park then used the HAT domain, the histone acetyl transferase domain of CBP to acetylate the p53. And acetylation occurred, we can follow this by NMR, at lysine 372, lysine 373, and lysine 382. And upon acetylation, the chemical shift changes were reversed. So acetylation of these residues decreased the perturbation of chemical shifts in the presence of DNA. It abrogated binding of the p53 through the CTD to DNA. So to summarize that, we see that basic residues in the CTD undergo dynamic interactions with the DNA outside the cognate 20 base pair recognition element. The interactions are predominantly with this very basic region between about the residue 368 and 375. So we see the greatest shift perturbation for residues in this region. And the sites of acetylation are marked here. So the role of the CTD is to mediate, form these transient interactions with the DNA and mediate fast sliding on the DNA to facilitate the search for cognate p53 binding sites. Acetylation by a histone acetyl transferase modifies 372 and 373 and 382 and abrogates the interactions between the CTD and the DNA. And acetylation at these sites is actually required for activation of low affinity promoters that bind, that regulate pro-apoptotic genes. And by acetylating the CTD, the specificity for the p53 response elements is enhanced. This has been known for some time. It's too early in the morning for me. I've got a frog in my throat. Let me now switch to the N-terminal activation domain, the NTAD. And when Alex generated these full-length p53 constructs with the N-terminal activation domain segmentally labeled, he discovered something rather interesting. There were interactions between the N-terminal activation domain and the DNA binding domain in the full-length p53. And you can see this in an HSQC spectrum, where if you look at the cross peak of threonine 55, for example, it in black is the position of that threonine 55 in an isolated peptide, residues 1 to 61 peptide. In the full-length p53, segmentally labeled in the N-terminal region, that peak is shifted and weakened, and other peaks are also shifted and weakened, showing interactions between the NTAD and some other part of p53. The magnitude of the chemical shift perturbations are small in the AD1 region, much larger in the AD2 region, and there are also changes in intensity of the cross peaks and peak height. Particularly in AD2, the peaks become weak and broad. AD1 is affected to a lesser extent. If one adds DNA, then these chemical shift perturbations are reversed. So here's T55, for example. There's the peptide, free peptide. There is full-length p53. Upon addition of DNA, the peak starts to shift back towards the free position. Likewise for here for L45 and other residues. So in other words, the DNA is competing with the NTAD for binding to the DBD. The N-terminal activation domain is able to make transient interactions with the DBD that can be competed off by DNA. So to confirm that the interactions were with the DBD, Alex spin labeled the DNA binding domain, putting a spin label at residue 121 close to the DNA binding surface, and then looked for perturbations for broadening of resonances in the N-terminal activation domain. And he sees significant broadening in the AD2 region, somewhat less broadening, a smaller PRE in the AD1 region, indicating that the interactions between the N-terminal activation domain and the DBD are close to the DNA binding surface or in the DNA binding surface, and the AD1 region is making lesser interactions than AD2. He also put spin labels into the N-terminal activation domain itself and looked at the reverse PREs, and with spin labels at a cysteine placed at residue 58, or also at S15, making that into a cysteine, you can see PRE effects on the DBD. And this just shows some of them from the spin label at 58. The magenta coloring shows where we see broadening of DBD resonances by the spin label in the position 58 in AD2. And you can see that this is close to and in the very basic DNA binding surface. And by looking at doing salt titrations, etc., Alex could show that these interactions between the N-terminal activation domain and the DBD were electrostatic in nature, primarily electrostatic in nature. So the question then arose, if the NTAD interacts with the DBD, does it impair DNA binding? And so Alex carried out fluorescence anisotropy titrations. And this shows the changes in fluorescence anisotropy upon titrating the p53 into DNA, 20 base pair DNA oligonucleotides containing the p21 recognition sequence or a non-specific DNA sequence. You can see that the red and the black curves, black is full-length p53, red is p53 with the N-terminal activation domain deleted. So it's the construct that consists of residues 62 to 393 no N-terminal activation domain. And you can see that in the case of the non-specific DNA, once the N-terminal activation domain is deleted, the p53 binds more tightly to the non-specific DNA. Here are the KDs obtained by fitting the data. The N-terminal activation domain makes very little difference for binding to a specific p21 high affinity p21 sequence, but for non-specific DNA, the N-terminal activation domain, by dynamically interacting with the DBD, substantially weakens, a factor of about 6, the binding affinity for non-specific DNA. And so it's increasing the specificity for a specific P21 recognition element. The specificity for binding to a recognition P21 element, if the NTAD is missing, is 4 compared to a factor of 23 in the presence of the NTAD and the full-length P53. So this led us to a model for how the NTAD, the N-terminal activation domain, and the C-terminal regulatory domain function synergistically to enhance specificity for DNA of P53 regulatory elements and facilitate a rapid target search. So in the absence of phosphorylation or any other post-translational modification, the NTAD is dynamically interacting with the 4 DBDs of P53. So inhibiting binding to non-specific DNA. The tetramerization domain presents the 4 CTDs to the DNA. They're interacting outside the DBD binding site dynamically and holding the P53 weakly onto the DNA. This enables rapid diffusion along the DNA until, or jumping, etc. as well, until a recognition element, a specific recognition element is encountered. When the P53 encounters a specific recognition element, now the binding to DNA is more favorable than binding to the NTAD. And so the NTAD is released, is then available for binding to the coactivators, to CBP and P300. So we can get interactions with CBP and P300 that acetylate the CTD and prevent it from making further interactions with the DNA. So in this way, we get rapid search of the DNA through interactions with the CTD, with non-specific DNA. The DBD binding is weakened by interactions with the NTAD. At a recognition element, this whole process is reversed, and the specificity and affinity for the specific recognition element is greatly enhanced. So this shows how the CTD and the NTAD can function together with the DBD to promote specific binding and facilitate diffusion on the DNA. So what about threonine 55? I mentioned that earlier. This is one of the known phosphorylation sites, but not much was known mechanistically about how it functions. And this is all the rest of the work I'll talk about is by a postdoc in the lab, Chan Sun. So what was known from the biology was that threonine 55, which is in the AD2 region of the transactivation domain, threonine 55 is constitutively phosphorylated when cells are unstressed. After DNA damage, P53 is activated by dephosphorylation of T55. Towards the end of a stress response, DNA damage response, T55 becomes rephosphorylated, and this leads to termination of the stress response. There's the references for, but discuss the biology behind this. And so Shan was very interested to understand how does phosphorylation of T55 in this NTAD modify the behavior of P53 towards DNA binding. Now, firstly, we had to generate a construct that could be specifically phosphorylated at T55. Well, it turns out that CK2 is very efficient at phosphorylating T55, but using HSQC spectra, we could see that CK2 also phosphorylates serine 6, serine 46, and serine 392. And so we needed to make mutations in P53, full-length P53, again using the superstabilized variant, so that we could achieve specific, site-specific phosphorylation. And I'll talk about two constructs. One I'll call, you'll see written as T55 P53A3. That contains 3 alanines, S6A, S46A, and S392A. So in the presence of CK2, only T55 is phosphorylated. The other construct contains only 2 alanines, 6 and 392. So CK2 phosphorylates completely at 46 and T55. So we get a doubly phosphorylated P53. So upon phosphorylation of T55, P53 is actually released from a recognition element. This shows fluorescence anisotropy changes in the presence of CK2, where the T55 P53A3, so there's only one phosphorylation site, where it's bound to an apoptotic promoter, the DNP1 DNA recognition element. And so with no CK2, following the changes in anisotropy over a couple of days, there's no change in anisotropy. In the presence of CK2, the anisotropy rapidly decreases because the P53 falls off the DNA. This would be this dotted line here indicates how the anisotropy would differ between the DNA bound form and free DNA. And so over a couple of days, the P53 completely falls off the DNA. So we turn to NMR to understand what the phosphorylation was doing to the interactions, the transient interactions between the N-terminal activation domain and other parts of the P53. And what we found was that the interaction between the AD2 motif of the N-terminal activation domain, and remember this is where T55 is, so the interactions of that AD2 motif with the DBD are enhanced by phosphorylation of T55, and they're further enhanced by phosphorylation of both T55 and serine 46. So you can see this in the spectrum. The tryptophan indole resonance has turned out to be a really nice indicator of binding to the DBD. So tryptophan 53 is shifted when the protein interacts with the DBD and it becomes weaker. You can see that the shift is a rather large shift on phosphorylation of T55. It shifts even further when both are phosphorylated and becomes weaker. And there's no change in the TRP23 peak intensity or chemical shift. So this acts as a nice normalizing probe. But you can see that the intensity of the tryptophan 53 resonance decreases upon phosphorylation of T55, further decreases on phosphorylation of tryptophan of serine 46, showing that the interactions with the DBD are becoming enhanced. If you look at the average intensity of AD1, no change upon phosphorylation, whereas the average intensity of AD2 decreases significantly, AD2 resonances, upon phosphorylation of 55 and or 46. We also see broadening of resonances in the PRD, proline-rich domain, when both of these residues are phosphorylated. So from the changes in chemical shift and cross-peak intensity, we can see that the interactions are enhanced. We also see it from PRE experiments, putting a spin label at 121 on the DBD again. This shows a difference in the PRE between the phosphorylated protein, phosphorylation at T55, and the non-phosphorylated protein. And these negative peaks show an increase PRE in the phosphorylated species. So you can see that the AD1 is actually released from the DNA upon phosphorylation of T55. The interactions with AD2 are enhanced, and interactions with the proline-rich domain are also enhanced by phosphorylation of T55. So what does it do to DNA binding? So Sean measured the binding affinities using fluorescence anisotropy with Psi5 labeled 20 base pair oligos on a P21 recognition element, which is involved in cell cycle regulation and is a high affinity binding site, and DINP1, which regulates apoptosis and is a low affinity site. So you can see that without phosphorylation at either 55 or 46, we get a normal binding curve. Binding's weaker on the DINP1 site, but again, it's a normal binding curve. Upon phosphorylation of 55 alone in black, or both 55 and 46 in red, we see a bell-shaped curve, a very surprising bell-shaped curve, which indicates auto-inhibition. As the P53 concentration is increased, then the protein first starts to bind, but then it inhibits, auto-inhibits, and reduces the binding. And that's consistent with what we saw in the previous experiment I described, where phosphorylation causes the P53 to fall off the DNA. You can see that the effect's even more pronounced with the DINP1 site, where essentially binding is abrogated upon phosphorylation of 55 or 55 and 46. So the model that Sean came up with to explain this is shown here. And he was able to numerically fit those curves, the fitted lines you can see there. You could fit the curves using these models. So firstly, I remind you that P53 binds as a dimer of dimers to two 10 base pair half sites in 20 base pair oligos. So we've got two specific sites, half sites in a 20 base pair oligo. So binding of non-phosphorylated P53 has been measured by many people, and it's highly cooperative with the Hill coefficient of roughly 2. So that's a simple process that's well understood. In the presence of threonine 55 phosphorylation, the mechanism is as follows. A dimer of the phosphorylated P53 binds to the DNA, followed by a second dimer. This binding process is non-cooperative. The phosphorylation completely eliminates the cooperativity of binding of the phosphorylated P53. The Hill coefficient is approximately 1. When the second dimer binds, we get auto-inhibition and the complex dissociates. So we can describe with KD2 the auto-inhibition, KD1 the binding event, and the dissociation process is highly cooperative. The Hill coefficient is between 3 and 4. This is actually some numbers. And there's two things I want to point out here. Firstly, the binding event, this is on the P21 promoter, or the DINP1 DNA. Firstly, as the protein becomes phosphorylated at T55, firstly, the Hill coefficient goes from 1.9 to 0.9. Secondly, the KD increases. Binding becomes weaker upon phosphorylation of T55, weaker still upon phosphorylation of both T55 and S46. You see the same trend for DINP1. The delta G for binding changes by about 0.6 kcals per mole per phosphoryl group. And that's interesting because we've seen that number before. Looking at, we see in interactions of P53 with domains, TAS1 and TAS2 domains and other domains of CDP, upon multi-site phosphorylation, we see the same number, that the increase in affinity in that case upon phosphorylation follows 0.6 kcals per mole per phosphoryl. But this is going the other way. As you phosphorylate at multiple sites, you enhance the binding affinity. In the case of the DNA binding, as we phosphorylate at these two sites, we decrease affinity through the phosphorylation, but the delta delta G is the same. And similar numbers have been seen for other systems other than P53. Turning to the autoinhibition, the KD2 is not particularly well defined. I've just given approximate numbers there as an average, but it's roughly between about 110 and 150, 160 nanomolar. And now the important thing is that the Hill coefficient for binding of the PT55 phosphorylated P53 is about 4. It's 3 for binding of the doubly phosphorylated. So we've got a highly cooperative dissociation process. Okay, what's the proline-rich domain do? What's its role in this? Well, Shun decided to delete the proline-rich domain, shown here. So the internal activation domain is directly linked to the DNA binding domain. So what happens? The interaction becomes stronger. So you can see it here. Here is a 1:95 peptide. So there's no DNA binding domain. There's a nice intense resonance from T55 or PT. This is not phosphorylated now, it's just 355. So in the full length P53, you can see the resonance is shifted to the blue peak here. Deletion of the proline-rich domain shifts it further and weakens it further. And these resonances all lie on a straight line. And the same is true for other resonances that we can follow undergoing shifts between the peptide, the full-length P53, and the delta PID peptide are all in a straight line, showing this fast exchange between the interactions between the NTAD and the DBD. Now, what about DNA binding? So binding to either the P21 element in blue or the DINP1 element in red, this is now in the absence of phosphorylation. So even without phosphorylation of T55, there's autoinhibition of binding to P21, and even greater autoinhibition for binding to DINP1. So we get autoinhibition in the absence of phosphorylation, whereas with the PID present, autoinhibition only occurred upon phosphorylation. If T55 is phosphorylated, the autoinhibition becomes even stronger because the interactions with the DBD become stronger. So we have autoinhibition in the absence of phosphorylation. And this explains a puzzling observation from the literature from way back in the 1990s, and there were several papers that related to this, that a molecular biologist found that deletion of the PID abrogates P53-mediated activation of apoptosis genes, but had no effect on genes that regulate cell growth. And we can see here that the effect on a strong P21 promoter is minimal, but on an apoptosis promoter, deletion of the PID has a dramatic effect in terms of autoinhibition. So it explained that, nicely explained that biology. Now, it turns out that you can actually describe this process as an equilibrium, the autoinhibition, in terms of an equilibrium between an open state and a closed state. Open, where the NTAD is free and flopping around. Closed, where it's interacting with the DBD and is unavailable, because the DBD is unavailable for binding of DNA. I've already mentioned that there's fast exchange between these O and C states, and I sort of alluded to it earlier without saying very much. We can actually measure the population of the open and closed states from the intensity of the tryptophan 53 and tryptophan 23, the relative intensity of the W53 and W23 indole NH cross peaks in the HSQC spectrum. And so this is a plot, a histogram showing the percentage of the closed state for the various constructs that we've looked at. So there's non-phosphorylated P53. There's the double mutant with S56 and T55 present, but the 2 alanine is mutated. So this is unmodified P53. Phosphorylation of P53, a substantial increase in the closed population of closed state. Phosphorylation of, I'm sorry, that was the delta PID, I'm sorry. So deletion of the PID itself is enough to substantially increase the population of the closed state. Phosphorylation of T55 also greatly increases the population of the closed state. The double phosphorylation of 46 and 55 increases it further, and phosphorylation of 55 and deletion of the PID leads to nearly 90% population of the closed state. Now, from this, of course, we can calculate free energy changes, and we see that for the deletion of the PID stabilizes the closed state by about 0.2, 0.3 kcals, et cetera, et cetera, et cetera. So you can get an idea of the free energy involved in the stabilization of this closed state for the various phosphorylation events or deletion of the proline-rich domain. One further point is that the strength of the interaction between that N-terminal activation domain, the AD2 region, and the DBD depends upon the number of DBDs. And we had a hint of this in the early experiments that I talked about with Alex Croyce. Alex, with the spin labels, was using just a monomeric P53 where it deleted the tetramerization domain in the CTD. And the interactions there were fairly weak. And we had a pretty good idea they were stronger in the presence of the 4 DBDs. And we can see that immediately here. If we take a dimeric P53 with the L344A mutation, then we see that the interactions of the NTAD with the DBDs is much weaker. So this is an intensity, relative intensity plot that's turned up the other way around from what you saw in the data from Alex at the beginning. So the blue histogram indicates broadening, weakening of resonances, less intense resonances in the phosphorylated T55 tetramer. And so a lot of broadening, a lot of weakening of resonances in AD2. In the case of the dimer in red, again phosphorylated at T55, you can see that the intensity changes are much less. The interactions of the dimeric DBDs with the NTAD are greatly weakened. And we can understand this as follows. In a dimer, we only have what I call cis interactions, where the AD2 region can interact with its own DBD or with the other DBD in the dimer bound to DNA. Assuming it's bound to DNA. This is just showing where the DNA would bind. In the tetramer, on the other hand, we get these same cis interactions, but we can also get trans interactions where AD2 in one of the activation domains on the other dimer interacts directly with the 2 DBDs in the other dimer. And if you look at the structures, the X-ray structures of the tetramer bound to DNA, you see that 2 of the NTADs would point directly towards the neighboring dimer. And so these would strengthen the interactions in the presence of the 4 subunits of the P53. Okay, so to summarize, perhaps I should add that this explains also the auto-inhibition. The P53 binding first to DNA as a dimer can still bind fairly tightly. There is some competition, so we weaken the binding, we lose cooperativity. When the second subunit binds, the second dimer binds, then we get these enhanced interactions that lead to competition with binding to DNA. So to summarize and bring it back to the biology, threonine 55 functions as a master regulatory switch that is involved in activation or termination of the P53 mediated stress response. So in an unstressed cell, T55 is constitutively phosphorylated. What that means from our data is that will abrogate binding of P53 to its target sites and also to non-specific DNA at the low concentrations of p53 in an unstressed cell. So the p53 just won't be able to bind and will fall off the DNA. This prevents aberrant activation of p53 target genes in an unstressed cell. The cell doesn't want to have the damage response, DNA damage response turned on when there's no DNA damage. And so this prevents aberrant activation. Again, by releasing the, or abrogating the DNA binding, p53 is released from the anchoring DNA. It's free to float around. And as a consequence, it's exported to the cytoplasm. This was the biology again. This was known for a while, that in an unstressed cell, p53 is exported to the cytoplasm and it's degraded. After genotoxic stress leading to DNA damage, threonine 55 is dephosphorylated by a protein phosphatase, the protein phosphatase PP2A. This is activated, so in response to DNA damage, there's a phosphorylation cascade. The ATM kinase phosphorylates serine 15 and other kinases in other serines in P53 to activate it. It also phosphorylates a subunit of PP2A to activate PP2A, which then dephosphorylates T55. So upon dephosphorylation, what we know from our experiments is that will weaken the interactions with the DBD, between the DBD and the NTAD, and relieve the autoinhibition of DNA binding. So that activates P53 for binding to P21 and to all of its other target promoters. So after the DNA damage response has gone on for a while, it has to be turned off. How is it turned off? How's the DNA damage response terminated? It's terminated because one of the transcriptional accessory factors, TAF1, associated with the polymerase initiation complex, rephosphorylates T55. So upon rephosphorylation, then the NTAD of the phosphorylated PT55 phosphorylated P53 will fall off the DNA and terminate the DNA damage response. So that rephosphorylation will shut down the entire process. So that brings me to the end of my story. And what I want to stress is that what I've shown you is that the intrinsically disordered regions and the structured regions, particularly the DBD of P53, function synergistically to regulate the DNA damage response. If one looks at these in isolation, you don't get the story. You need to study the full-length protein. And by using these sort of powerful NMR techniques and other biophysical methods to look at the full-length protein, we can begin to get a complete picture of the very complicated regulatory processes. And obviously P53 is just one example. As I said, well, the majority of eukaryotic proteins, human proteins, contain both ordered and disordered regions. And so we need to understand how these regions can synergize to regulate the function of these very important regulatory proteins. So I've acknowledged most of the people as I've gone along. Alex Croix, Shan Sun, Josephine Ferrion, Sang-Ho Park, Gabriel is currently working on some other questions related to full-length P53. Maria Martinez-Yamut plays a critical role in almost everything we do. Maria is responsible for designing INTEAD constructs and the various molecular biological constructs and figuring out how to express them at sufficient level and label them for the kind of experiments we want to do. So she plays an absolutely critical role. And of course, this has been a collaboration with my long-term collaborator, Jane Dyson, who's been involved in every aspect of this work. So with that, let me thank you for your attention, and I'm happy to take any questions.
Thank you very much, Peter, for this tour de force into the function and structure of P53. And perhaps I will take the prerogative to ask the first question. So it's quite amazing that as one phosphoryl group has such a dramatic effect on the biology of P53. What is it on the microscopic level that makes it make this dramatic effect? Is it just the charge, the added charge to the protein, or is it something beyond that?
No, it just seems to be added charge. So the phosphorylation is enhancing the electrostatic interactions with the DBD. And the critical thing is that there's an avidity effect there. The interaction with a single DBD is reasonably weak. Even upon phosphorylation, it's enhanced, but it's still relatively weak. You saw the delta G of 0.6 kcals. But when there are 4 DBDs present, then a single NTAD can interact with all 4 of them. So we get these dynamic interactions between the 4 NTADs and the 4 DBDs. And so the effect is amplified by having multiple subunits, multiple interaction sites on the 4 DBDs. We don't yet have a good picture of exactly where the NTAD's interacting. These are experiments that are still to be done, or but we know that the interactions are near the DNA, near or in the DNA binding surface, and they're predominantly electrostatic.
So you can fully reverse them by increasing the ionic concentration, for example?
Yes, yes. I should have mentioned that all of the DNA binding experiments were done at 150 millimolar salt to be close to physiological conditions. So it's still interacting there. But if one increases with an isolated monomer of P53, that NTAD DBD interaction is eliminated at about 300 or 400 millimolar salt. So you can back it off with salt. Now, we've not tried that with the full-length P53, but I'm sure it will also be salt sensitive to some extent.
Okay, thank you. So we have a question from Ziad Ibrahim. Ziad?
Yes, okay. Hi, thanks for the talk. I was actually wondering if you measured the KD for the interaction between the NTAD and the DBD when the NTAD is phosphorylated or not phosphorylated.
No, we've not. We would be doing that using isolated peptides again, and we have not done that. But we have an indication of the strength of the interactions from the delta Gs.
Okay.
Yeah.
So you think it's lower? It's lower than the affinity for the binding between the DBD and the DNA.
Yes, it is, but it's an intramolecular effect, and as I mentioned in response to Gilad's question, there's an enhancement by having 4 DBDs present. Mm-hmm. But we don't actually have an affinity. We've not measured that, and it would be using isolated peptides. Of course, you wouldn't get the correct affinity because as you see with the removal of the PRD. The linkage between the DBD and the distance between the DBD and the NTAD is critical in terms of determining the affinity.
Yeah. Thank you.
Okay, Phil Selenko.
Yes, hey Peter. I thought this was super exciting, and I think it's even a broader context, because what you just showed us is that cell signaling at a single phosphorylation site has a structural component, because it does lead to some sort of, intramolecular looping out and re-engagement of a faraway site with the DNA binding domain. And when it is phosphorylated versus, opening it up again when it is unphosphorylated. And I think this is an amazing concept that is probably present at many of those disordered regions, that phosphorylation can really completely rearrange a disordered N-terminus. I don't know if you would agree with this, but, for me, this was a stunning example of such an effect. My question is, what is special about, T55? Is it just closest to the DBT, and that's why you think you get this, highly unusual sort of reverse behavior for T55 and not any of the other phosphorylation sites? So what makes it special?
Yeah, that we don't know. So firstly, I agree entirely about your first comment about this intramolecular effect, the regulatory effect of a single phosphorylation. And I think this is exactly the point I was trying to make: the synergy between the disorder and the ordered regions, and how these are present in the majority of eukaryotic proteins. We have to look at the full-length proteins to understand how these processes work. We've just missed them. You can't see them if you use isolated domains. So coming back to your question, so yeah, we don't know what's special about it. So the AD2 region is closer than AD1 to the DBD. There's a lot of negative charge around this, and promotes the interactions with the basic surface. And adding that extra charge is clearly important. There may be interactions. There's a tryptophan at 53, for example, and a phi at 50. There may be some hydrophobic content to the interactions as well. These are things that we need to understand. But we know that the AD2 region does dominate the interaction with the DBD, even in the absence of phosphorylation. It's stronger than it is for AD1. AD1's further away, so there's a larger entropic component there, of course. But we still have to really try and dig in and find out exactly what that T55 phosphorylation is doing. There's nothing that stands out from the sequence or from anything except proximity.
Okay, Sandhya Bhatia wanted me to read their question. Is the isomerization status of peptidyl-prolyl bonds in the proline-rich domain important for its role? Or is it simply acting as a stiff linker to prevent the interaction between its two adjacent domains?
That's not entirely known. There is a phosphorylation site at 81 in the PID, proline-rich domain. And there are binding sites for proteins. Some of the polyproline regions in there can be recognized by SH3 domain, but not much is known about its role. It's probably just a linker. It certainly has to be there. If you take it out, as we know, then P53 is auto-inhibited. So it's playing an important role as a rather stiff spacer, but there are other events can occur there that aren't yet understood. The T81 phosphorylation, what it does, we just don't know. And it's been a region that's been poorly understood ever since P53 has been studied. There's a lot of speculation, but not much is known about it.
Okay, Ben also wanted me to read this question. These are wonderful insights on P53 function. Is there more detailed information about the sliding process of P53 before reaching the recognition element, e.g. speed of motion on DNA and time that it remains bound before dissociating?
Yeah, there is a lot of information there. So several groups have studied the sliding of P53 on DNA, the search for target sites using SM-FRET, etc. And there's a big literature on that. A lot is known that the search process is very important. The only thing that we've added is understanding about where the CTD interacts with the DNA and the synergy between the CTD and the N-terminal activation domain in terms of lubricating the sliding process. But the fluorescence techniques have actually put numbers on residence time, sliding rate, etc. If you're interested, I can send you the references, but there's several papers on that.
Okay, next one, Laila.
All right, thanks, Gilad. First of all, Peter, that was absolutely lovely. And you've made such a case for the need to go beyond domain dissection and domain-domain interaction. One needs to look at the whole beast, which is terrific. I guess I was curious because there's so many disease-associated mutations known in P53, and unless I missed it, you didn't tell us how they may relate to some of these interactions that you've been talking about. I know they're hotspots, et cetera, but do they play in? Because obviously it's a disease propensity, so the networks still have to work. They just don't work properly. So you don't want to disrupt completely an interaction, but you would see it in some way be unbalanced. So I'm just curious about that.
Yeah, most of the mutations, the disease mutations, are in the DBD. And they either destabilize the DBD and lead to aggregation, phase separation, as recent suggestions that a phase separates, and other mutations in the DBD impact the DNA binding. There are very, very few pathogenic mutations known in the N-terminal activation domain. And I must confess, I've not looked at them to see whether they would interfere with this process. That's a great idea. I should go back and do that. But there's only, out of the hundreds and hundreds of mutations known in P53, there's only about 5 or 10 in the N-terminal activation domain. And they're probably too critical. If you make mutations there that stop it binding to co-activators, or it's going to mess up the whole process.
Yeah.
They'll be lethal.
Okay, thanks.
Okay.
Well, thanks, Peter. That was really beautiful talk and very convincing thermodynamic analysis. Actually, I was wondering about the dynamics. So once you have the equilibrium of open-close states of the N-terminal domain binding to the DBD, I was wondering whether the NMR experiments would also allow you to sort of get the kinetic rates of switching between open and close, and maybe if the N-terminal domain is bound to the DBD, could it serve as a sort of lubricant for sliding on the? You mentioned that already in your response to another question. So, is there any information about rates?
No, there isn't at the moment. No, there's a vague information about rates, so we know it's fast. So it's fast on the NMR chemical shift time scale, and so the resonances are shifting in a linear fashion as that interaction becomes strengthened.
Mm-hmm.
And so fast on the NMR timescale means it's probably on a millisecond or faster timescale.
That would mean it should be accessible to CPMG experiments.
Absolutely, yeah. This is not something we haven't done that yet, but that's something that's definitely in mind, because we are seeing broadening as those peaks shift, as they interact. And so, using CPMG experiments or CEST experiments, we should be able to pull out actual rates of that process, and that's definitely something in our to-do list. Yeah, so
single molecule threat. Again, you should put probes there and measure it.
Mm-hmm, mm-hmm.
Yeah, I think it would be.
I would go for CPMG.
But I think I'm not sure whether. So the Fred experiment should give some information about how that affects sliding. So, for example, if one was to take the T55 phosphorylated full-length P53, and where the interactions with the DBD are enhanced, then
I think so, yeah.
How fast does that slide by fluorescence compared to the non-phosphorylated, or one where the NTAD has been chopped off?
Yeah.
So I think you could get direct measurements about sliding rate and the lubrication, if you want, from fluorescence measurements.
Oh, I would think so too. Okay, exciting. Thanks, Peter.
Okay, it looks like there are no further questions. So I would like to thank you again, Peter, for this really terrific talk. And I would like to thank you all for staying till the end of this session. And please come back in 2 weeks for a talk by Victor Munoz. So thank you all, and good night, or good morning, or good afternoon, or wherever you are.
Good morning.
Thanks a lot, Peter.
A pleasure.
Bye, everyone.